Hola a todos, ¿cómo están? Muy buenos días. Welcome to the Symposium on Neurodegeneration and Aging. Today we have fantastic speakers, Dr. Gonzalo Arboleda, Dr. Luis Barbeito, Andrea Paula Lima y Dr. Andrea Paula Lima y Dr. Lionel Miller Igas. We are going to present the Dr. Gonzalo Arboleda. He will not to be with us today, but Dr. Adrian Sandoval will be with us discussing and answering the questions. Dr. Gonzalo Arboleda is researcher at the Genetics Institute of the National University of Colombia, full professor at the Medical School of the National University of Colombia, PhD in neuroscience from the University of Manchester, postdoctoral fellow, Medical University of South Carolina, Children's Research Hospital in Developmental Neurogenetics. And today he will going to present the conference named Nuclear Receptor Potential Therapeutic Targets for Alzheimer's Disease. Welcome and enjoy it.
Faculty of Medicine at Universidad Nacional de Colombia. I want to thank the organizers for the kind invitation to speak today. Today I'm going to talk about a recent work on the role of nuclear receptors as a potential therapeutic alternative for Alzheimer's disease. The content of my talk will be divided into points. One, the general view of Alzheimer's disease, and second, some of our recent results on the role of LiverX receptor in Alzheimer's disease. So as we all know, Alzheimer's disease is the main cause of dementia with a big social and economic burden. As we can see here, the numbers are increasing year by year, with increasing also in the budget of this for the treatment and care of these patients. However, there's no treatment that blocks the progression of the disease. From the pathological perspective, Alzheimer's is characterized by the accumulation of senile plaques, extracellular senile plaques, mainly composed of amyloid beta, and intracellularly by the accumulation of hyperphosphorylated tau in the form of neurofibrillary tangles. However, there are additional features of Alzheimer's. In particular, in neurons, there's clearly a synaptic dysfunction. Also, in astrocytes and microglia, there's a pro-inflammatory phenotype, decreasing in growth factors. There's also damage in blood-brain barrier, disruption of the blood-brain barrier, and also changes in oligodendrocytes that cause demyelination. So this makes Alzheimer's a very complex disease. The main hypothesis around Alzheimer's disease is the amyloid cascade hypothesis that describes the metabolism of the amyloid precursor protein by two pathways, the non-amyloidogenic pathway by the cleavage of by alpha and gamma secretase, and the amyloidogenic pathway by cleavage of beta and gamma secretase that generates amyloid beta and the accumulation of this amyloid beta in the form of oligomers or plaques. The role of these plaques have been the main point of research around Alzheimer's, because these plaques, and in particular oligomers of amyloid beta, cause synaptic dysfunction, tau aggregation, inflammation, mitochondrial damage, leading all these to neuronal death. So the amyloid hypothesis has been the main target for the therapeutic alternative. So the therapeutic alternative based on the amyloid hypothesis has been developed mainly 2 points: blocking the amyloid beta production by blocking the role of these secretases, and by blocking the aggregation of the amyloid beta once it is produced. And the second alternative is to increase the amyloid beta clearance, either to degradation or through release of amyloid beta through the blood-brain barrier, or transported to microglia and astrocytes and be degraded by these two cells. The particular emphasis of these beta clearance alternatives has been the anti-amyloid immunotherapies. However, many of these alternatives have been show no clinical benefit despite reducing the levels of amyloid beta. Recently, one of these anti-amyloid beta immunotherapies, a monoclonal antibody known as aducanumab, has been approved by the FDA for the treatment of Alzheimer's disease. However, there's a lot of controversy around this at the moment that I'm not going to talk about. Additional alternatives, in particular, are associated to the role of ApoE. So the main source of ApoE in the brain is astrocytes. So when ApoE is produced, it can be lipidated through this transporter, also present in astrocytes, ABCA1. And once ApoE is lipidated, it can bind amyloid beta and increase the A beta clearance through the pathways that I just mentioned, through the blood-brain barrier, or be taken up by microglia and astrocytes for degradation. The important point here to keep in mind for what I'm going to show later is that the main regulator of ABCA1 and ApoE in astrocytes is the activation of nuclear receptors, in particularly the liver X receptor. So the point of our analysis was to try to analyze what is the role of this activation of this liver X receptor as a potential alternative for Alzheimer's disease. This is a general view of how liver receptors work. Once the ligand binds to the nuclear receptor which is bind to the DNA, a repressor is released and a co-activator is recruited. And there's activation now of the receptors that increase the production of the target genes, in particularly ApoE and ABCA1, but many other genes are also regulated by these nuclear receptors. So the question again is how the activation of the receptors using specific ligands could impact on Alzheimer's physiopathology. To answer this question, we use an artificial agonist, a synthetic agonist, GW3965, and we use three models: treatment of the triple transgenic mouse model, treatment of the hippocampus lysis with amyloid beta in the presence of GW, or hippocampal cultures treated with oligomers of amyloid beta, also in the presence of GW. And then we look for different changes in these three models. So initially what we found is that GW improves cognitive function, and this could be seen here, how the triple transgenic mice never learns to get to the platform in the Morris Watermaze paradigm. Here, sorry, this is the triple transgenic, and how the treatment with the agonist clearly improves this recognition of the platform and reduces the latency to platform in the Morris Watermaze. So this is the triple transgenic, and how the treatment improves to a level similar to the wild type, the latency to platform. Of course, we look for the expression levels of ApoE and ABCA1. So what can we see here is that treatment with GW in the transgenic, in the cortex and the hippocampus, increases the level of ABCA1 and ApoE. And this is quantified here. So the role of LXR is clearly increasing these two important genes for ApoE function. Then we look at the pathological changes in these mice, and interestingly, what we see is that GW doesn't change the A beta and tau load. This was made by immunohistochemistry and by ELISA, looking for A beta 40 and A beta 42. We didn't see changes in the hippocampus or in the cortex, and neither there was significant changes in the load of tau. However, there's a tendency to decrease in these levels of tau and ApoE in the brain of these transgenic mice. So the question was how the LXR agonist is improving the cognitive function in these mice if they're not changed in these pathologic hallmarks of Alzheimer's. So we look for additional features. We look for synaptic function particularly. So what we see here is the analysis of dendritic spines. And we see here that treatment with amyloid beta decreases the number, the density of spines, and how the agonist is able to increase this density of spines. But more importantly, the treatment of amyloid beta decreases the mature form of the spines, which are the mushroom spines. It is decreasing in a very significant way, and how the treatment with GW increases these mature spines. There's also an increase in the number of synaptic contacts, also analyzing different markers of pre- and postsynaptic proteins. So the conclusion from this part is that GW is able to increase the maturation of spines and increase the number of synaptic contacts. We then look at the electrophysiological potential of GW. What we see here is the LTP analysis, the control here in black, how amyloid beta is doesn't induce a LTP, but when we pretreat this, the hippocampus lysis with GW, there's a recovery of this LTP in a very significant manner. And then we did some analysis to look for the methylation of different genes. And what we found was that GW is able to differentially methylate synapses-related genes, in particular synaptophysin, in this case, which is shown here, how the triple transgenic decreases the expression of synaptophysin, and how the GW is able to increase, again, the expression of this synaptic protein in a very significant way. This doesn't happen with the PSD95 postsynaptic protein. So GW is able to change the expression and methylation of different genes, many associated to synapses. We only show here synaptophysin, but it's able to change many other changes associated to this synaptic function. Then we look for additional changes. In particular, we were interested in looking for activation of astrocytes. And what we see here is how the GW decreases the activation of astrocytes measured by GFAP, and also decreases the activation of microglia. This is the triple transgenic, and this is the triple transgenic treated with GW. And what we see here is the decreased expression of IBA1 as a marker of activated microglia. And we also look at myelin proteins. Here's an example of MVP, the myelin basic protein, how in the triple transgenic, there's a decrease of MVP, and the role of GW increasing the myelination of these areas in the hippocampus associated to increase expression of the myelin basic protein. We also look or try to look at the role of GW in the capacity to increase the proliferation of neuronal stem cells. And what we see here is that the triple transgenic mice, when we stain for ApoE and nestin, there's a decrease in the ApoE staining and let's say a basal staining for nestin. But when we treat with GW, there's a big increase in the marker for ApoE, but also a big increase in staining for nestin. And this was associated to an increase in the phospho-H3 histone, which is a marker for proliferation. So in conclusion from this part is that GW increases the proliferation of neuronal progenitors in the hippocampus and the subventricular area, and associated to increase in the proliferation of these progenitor cells. Of course, what we use to this point is a synthetic agonist, which is from a pharmaceutical company. So we wanted to look what is the capacity of plant extracts from the different areas of the country of Colombia. And we have a collaboration with a lab in the chemistry department that looks and analyzes plant products. And we tried different plant extracts for the LXR activity. We tried 82 extracts, and we use a luciferase assay to look for the capacity of this extract to activate LXR. And what we see here is that this is the positive control using GW. And what we see is that many of these extracts have a positive capacity to activate LXR. So we choose some of these extracts, and we did some experiments treating mice with these extracts. And what we see in these mice, the triple transgenic mice treated with extracts, is that the extracts increase ABCA1 and increase ApoE, very similar to what GW does. They recover the cognitive function of these mice in a very significant manner. They also decrease, and this is a very different result from what we see with GW, is that the plant extras were able to significantly decrease the level of amyloid load. What we see here is the triple transgenic, and this is the triple transgenic treated with the plant extract. There's a big decrease in amyloid load. And there's also a big decrease in neurofibrillary tangles. This is the triple transgenic untreated, and this is treated with one of the extracts. And what we see here is the big decrease in the intracellular neurofibrillary tangles. We also look for astrogliosis, and we see that the plant extract decreases very significantly also the level of staining with the GFP. There's also decrease in the macrogliosis measured with EVA1 staining in different areas of the hippocampus. And also what we see is an increase, a very significant increase also in the staining for myelin basic protein, suggesting a recovery of the myelination. So in summary, what we have shown is that activation of LXR, either with a synthetic agonist or with a plant extract, impacts in different points in the changes associated to Alzheimer's, particularly decreases amyloid and tau, it decreases inflammation, improves the synaptic function, improves myelin recovery, and increases proliferation of neuronal stem cells, Therefore, this activation of LXR, it has a potential multi-target alternative for or against Alzheimer's disease. And with this, I want to finish and thank the people who've done the work, many students from the neuroscience and cell-death groups, our collaborations, our funding sources. And thank you all for your attention.
Muchas gracias al doctor Gonzalo Arboleda. Sorry, I speak in Spanish. Thank you to Dr. Gonzalo Arboleda and Dr. Adrián Sandoval for this presentation. We have some questions on the chat. One is, which other genes are regulated by LXR that could be also, sorry, wait a moment, that could be also affected and can produce worse effects than the advantage in the cognitive function you got? Dr. Sandoval.
Thank you. Nuclear receptors are involved in the regulation of thousands of genes because there are the way that hormones works. Nuclear receptors works because they are transcription factors that interact directly with the DNA, the gene regulator zone of the genes. And different studies show that but the problem of this therapy is that it's difficult to control the interaction with these hormones with the other parts of the body. Different organs of the body will be affected by these hormones. And this is the difficult part to control here. But we believe, despite that, the cost benefit of this therapy will be benefit at the end to these patients than the problem that they could produce, if I understand the question.
Okay, thank you very much. There are other questions by Florencia Arredondo. Thank you very much for your talk. Could it be possible that you are seeing a synergistic effect of these drugs beyond LXR activation? Synergistic effect of the struts.
Of the what?
Struts, plants,
okay.
struts.
Yes, of course. We characterize these extracts in different ways. We also look for antioxidant effect of the extract, and also we characterize the inhibition of acetylcholinesterase in the extract, and other effects. We also studied the neuroprotection that can produce the extract when we expose the cells, the neural cells, to different neurotoxins like ceramide, glutamate, and amyloid beta, and others. And we look for the best extract that we found in plants in Colombia. We found that the extract has different activities, and we also believe that the extract produced a cumulus of effects in the brain of these mice.
Okay, thank you. We have a previous question from Diana Jerusalinski. Very good talk. Congrats. What about different alleles of APOE?
Okay. Recently was a study about a drug called bexarotene. Bexarotene is an agonist of the LXR nuclear receptor. Nuclear receptors of class 2 act on function or as heterodimers of LXR. That means that LXR works or function in a couple of LXR, and that means that if you activate LXR or LXR, you may produce a similar effect in the brain. And also bexarotene increase much more the expression of ApoE because in the ring regulator zone of ApoE, there are different response elements for different nuclear receptors, including LXR and PPARs. That means that LXR is able to produce or increase much more the ApoE. This work was proved in humans recently, and you can find this research in the ALS forum if you could read that. And they show it that those drugs produce a beneficial effects in patients when patients doesn't have the ApoE4 isoform. Okay, it may be because okay, it's difficult to explain that, but I think it's because ApoE4 is metabolized in different ways in the brain, maybe produce fragments of this protein that are toxic in the brain, and that way is because maybe this target is not working in these patients. But I believe the result was very promisorious in the other patients that was not ApoE4.
Adrian, we have no more questions on the chat, but I will ask you, what do you think about the regulation of lipids metabolisms, mainly the transcriptional genes regulations related with the enzymes of the lipid metabolisms by the inhibitor?
I don't understand.
Don't worry. Do you know if the inhibitor could to have any action on the enzymes, on the genes, on transcription of the genes related with the enzymes of lipid metabolisms?
Okay.
Because you could see that increase the ApoE in neurons, in new neurons, increasing the neurogenesis. But you attribute this part of the action to the hormones. But do you think that maybe part of the lipid metabolisms is related to?
Yes, of course. One of the main changes in Alzheimer's brains is the accumulation of the dysregulation of lipids. Also, cholesterol is increased in Alzheimer's brain and other toxic lipids. Why is that? We don't know really, but there are two different nuclear receptors involved in lipid regulation. One of these is the PPARs, alpha and gamma, and the other is the LXRs. LXRs regulate sterols, and PPARs regulates mainly fatty acids. And those nuclear receptors are involved in the lipid regulations, are interact each others. We are in the way to study the metabolome of this, of the therapy, of the changes involved in the therapy. And we believe that there are changes in lipids because the therapy involves the remove of lipids and maybe the reorder in the brain. Not only, as Gonzalo told, that maybe because this therapy involves gene expression reprogramming that was lost in the course of the disease, in the chronology of the disease, and maybe that was the cause that those lipids was accumulated in this.
Perfect. Thank you very much. Okay, thank you, Dr. Sandoval. We continue with our next speaker. Our next speaker is Dr. Luis Barbeito. He is researcher at Institute Pasteur in Montevideo, PhD in neuropharmacology, College of France, head of neurodegeneration laboratory, and member of the International Network of Pasteur Institutes. Welcome, Dr. Barbeito. The title of his conference is Glial Cells, Senescence, and Neurodegeneration. Okay, enjoy it.
Good morning. I'm Luis Barbeito from the Instituto Pasteur Montevideo, Uruguay, and I would like to thank the organizer for this FALAN meeting. I'm very pleased to participate. So my subject today will be glial cell senescence and neurodegeneration. So cellular senescence is a recognized mechanism of aging. And the idea is that senescent cells, once appear, start to accumulate during aging and underlie the pathogenesis of multiple pathologies, including cancer, atherosclerosis, diabetes, immune dysfunction, inflammation, neurodegeneration, which is our subject today, as well as neurovascular defects, and so on. So, how senescent cells appear? So, aging is related to DNA damage, of course, accumulation of mutations, telomere attrition, is related to cellular stressors that can appear as oncogenic insult or reactive metabolites, intoxications, stress, also to other type of stress, or mainly oxidative stress and inflammation, as well as DAMPs. All of these will cause senescent signals that finally are detected by a number of sensors in the cell, such as P53, for example, that sense DNA damage, and will also trigger the expression of other anti-proliferation and anti-oncogenic genes like P16, for example, or P21, that will stop proliferation of the cell. Okay, and so the result of all these adaptive changes to damage, to cellular damage, will be the appearance of a cellular senescence. Of course, most of these cells will die by apoptosis or necroptosis and will disappear of the system, but those cells that survive will develop a characteristic phenotype that defines and characterizes the cellular senescence, and these cells become flat, enlarged, with, of course, they don't proliferate anymore, and they develop a number of morphological and functional features that are known as, for example, SASP, which is the secretory, the senescence-associated secretory phenotype, which is another feature of the senescent cells that will make these cells quite characteristic. I mean, they are functionally, morphologically, but also from the secretory aspect. These cells will be much more pro-inflammatory, pro-trophic, and pro-deleterious in some aspect to the cellular microenvironments, and as it's labeled here in this review article, cellular senescence has been described in most of neural cells in the central and peripheral nervous system, and could be involved in almost all neurodegenerative conditions. In particular, I would like to emphasize the SASP phenotype, the secretory phenotype, characterized by by cells that are enlarged with abnormal nuclear morphology, and that shows an increased activity of beta-galactosidase as a marker of increased lysosomes activity, altered mitochondrial activity, of course the expression of these anti-proliferation genes. These cells are characteristically resistant to apoptosis, and in the nuclei they have decreased levels of lamin B1 as well as high mobility box1, which characterizes phenotype. And from the secretory point of view, these cells will release a number of trophic factors that in some aspect are good for the cellular microenvironment, but also many pro-inflammatory cytokines as well as metalloprotease and other proteins that will alter the extracellular matrix. Okay, today I will focus on senescent astrocyte and senescent microglia. So in the case of astrocyte, it is known that in human beings and during aging, there is an increased number and accumulation of P16 positive astrocyte from the fetal stage to the aged state of the normal people. And up to 50% of the astrocyte in the forebrain could be a P16 positive cells, which means a huge amount. It is not known now whether these numbers represent the total amount of real senescent astrocyte, or rather P16 is expressed to push astrocyte to maintain a post-mitotic state. So this is an underlying discussion, but it's important. In Alzheimer's disease, this number further increase up to 50 to 60%, which represent a huge increase in the number of senescent astrocytes. So senescent astrocytes are characterized by a number of specific features. For example, the expression level of glutamate transporter is decreased, as well as the glutamine production. Mitochondrial dysfunction is very important, together with the production of reactive oxidative species, which will activate inflammatory pathways like NF-kappa B, and of course astrocytes are highly expressed the beta-galactosidase, which is an increased activity that characterize astrocyte in aging and in neurodegeneration. And also the astrocyte will release a huge levels of cytokines, inflammatory cytokines, that will alter the cellular microenvironment. Okay, in Alzheimer's disease, there is evidence that senescent astrocyte will release a number of mediators that will activate microglia together to induce neuroinflammation, as well as alter the blood-brain barrier permeability by metalloproteases that are released by senescent astrocyte, as well as TGF-beta, and at the synapses, it will also release different cytokines and inflammatory mediators that will disrupt the synapses and the synapses function. Okay, in the case of microglia, it is known that during aging, microglia will become dystrophic, and this is the particular morphology of microglia in aging, as compared with the normal microglia, with the teeny processes that move all the time, and in comparison with the activated microglia, the phagocytic microglia, that has this hypertrophic cell body and short processes, right? The dystrophic microglia is considered to be senescent cells, are characterized by being very active as pro-inflammatory, but also of having a downregulated phagocytosis and phagocytic activity. So senescent microglia together with astrocyte have been shown in a model of neurodegeneration of ALS, these ALS rats, by Emiliano Trias. And Emiliano found that both P16 positive astrocyte labeled with GFIP or P16 positive microglia labeled with EVA1 accumulate mainly in the symptomatic phase of the disease around the degenerating motor neurons, as you see here, and the numbers increase as the disease progression occur. So other markers of senescent cells as lamin B1 sharply decrease during disease progression. As you see here in red, the cells lose the lamin B1 marker in the nuclei. In this study, senescent microglia were also characterized in cell cultures by culturing or isolating microglia from animals in the end stage of the disease, where these microglia in culture become rapidly senescent after only 12 days in vitro, and these cells really express a number of markers, including beta-galactosidase, as is shown here. So in addition to glial cells, it has been suggested that neurons could also display markers of senescence, and in response to different kind of damage as stress, tau accumulation, beta amyloid, etc. So what is evidence indicate that these neurons could become senescent, although they are post-mitotic cells. They also can express upregulation of senescence markers and could also become SASP, that means senescence-associated secretory phenotype. So in this particular model, the senescent neuron related to A beta could become secretory and could induce secondary senescence to glial cells, and also neuroinflammation, as well as inducing neurofibrillary tangles in neighboring neurons. While the accumulation of senescent glial cells could induce neuroinflammation in neighboring glial cells, neurons, and vascular cells, the same is true, the opposite is also true. That means that neuroinflammation by inducing DNA damage and cellular stress could also induce further senescence in the glial cells. So also it has been suggested that damaged neurons, in this case, for example, having tauopathy, can become senescent-like cell, release a number of secretory factors, cytokines, among others, activate microglia and astrocyte, which in turn become senescent, and further can release secretory factors, senescent-associated secretory factors that will damage other neurons or the same neuron, aggravating the process. In this study, for example, Miranda Orr has used a pharmaceutical combination of dasatinib and quercetin that is known to specifically kill senescent cells, and she has treated animals having tauopathy with this combination of drugs, and she showed that the number of damaged neurons having a neurofibrillary tangle decrease by this treatment. Also, and also in the same study, she has shown that many functional items of these animals also improve by this senolytic treatment, deleting senescent cells in the dementia model. So in conclusion, we can say that the emergence and accumulation of senescent cells and the SAS phenotype allow a new understanding of brain aging. According to this hypothesis, the aging process could be modulated by targeting of senescent cells, for example, using pharmaceutical means and senolytic drugs. Neuroinflammation may be causally linked to the accumulation of senescent glial cells and perturbated by the SAS phenotype, of course. And finally, senolytic therapies might represent a new class of drugs for neurodegenerative disease. Okay, I would like to thank you for this meeting. It was a pleasure, and I remain available if you have any questions. Thank you.
Thank you, Dr. Barbeito, for your beautiful presentation and very interesting. I have not any question right now on the chat, but I have a question. Let me try to explain the situation. We have human brains in the group of neuroscience in Colombia. We are going to study this tissue, and we found that the familiar Alzheimer's disease produce astrogliosis, a lot of gliosis activations in the brain. But in comparison with the sporadic Alzheimer's disease, there are astroglial loss, several loss of astroglia. My question is, in this situation, in both situations, there are inflammation in the neurovascular unit involving the astroglia, the microglia, etc. But what do you think, in your expert opinion, is the main difference involving the aging and involving the degeneration mechanisms and this particular alteration supported in your hypothesis?
Okay, this is a very interesting question. Thank you. Well, I think that astrocytes must be considered as heterogeneous cells. I mean that they can adopt many different phenotypes and functions in any particular pathological situation. I would say that probably astrocytes are not the same from one patient with Alzheimer's disease compared to another, because they could be local differences and pathological difference. And so we must differentiate between a classical reactive astrocyte that are considered good and non-deleterious or pathological, which are very hypertrophic and responding to a damage, any kind of damage, exacerbating their normal function, with the other reactive astrocytes that are mostly deleterious and in shape they could be the same, but functionally are not the same. So there are subtle differences in astrocytes that make the difference, and so one would say that these deleterious or pathological astrocytes really go to the undifferentiation phenotype, right? They start to lose all the polar or specialization in different parts, one with the synapses, one with the blood vessel, and they start to secrete. They are very likely as a senescent associated phenotypic secretory phenotype, right? So we explore and we are investigating whether these deleterious astrocytes are in fact senescent astrocytes. And this is the point here.
Perfect. Thank you very much for your clarification. We have several questions, comments. Dr. Jaime Fornaguera, very interesting models. Congratulations. From Andrea, Cristina Paula Lima said, excellent talk. You, you showed that microglia is activated by the cytokines released by astrocytes, but could microglia instead sense pathogens and then activate astrocytes by releasing cytokines?
Okay, I think that there is a crosstalk between these two cells, and not only cytokines are involved. Cytokines are the most known mediators, but we must also consider a number of proteases and enzymes that are released that are very active, and they can activate many different pathways in pushing the inflammation.
Okay, next question is from Jose Góngora. Thanks, Dr. Barbeito, for your interesting talk. Is there any clue about the mechanisms by which quercetin kills senescence glial cells? Are there other phytochemical with similar effects?
Probably yes. Probably yes. All these senolytic drugs that kill the senescent cells are believed to function by pushing apoptotic pathways. So these cells are deleted by the system. Senescent cells have a very powerful anti-apoptotic mechanism. And quercetin is thought to act on this way, interacting with kinases and several enzymes. Quercetin is a multi-acting flavonoid, and many other flavonoids are just like quercetin. So we can expect that many other flavonoids could do the same and push toward the inducing apoptosis in senescent cells.
Do you mean that is removing the cell deaths, yeah, for improve the homeostasis, something like that?
Yeah, senescent cells, any kind of cells, they are resistant to apoptosis. They have developed, and so this is the reason they stay alive in the tissue. And so all the senolytic drugs, what they do is to push, to induce apoptosis in these particular cell types, not in others, eventually.
Okay, perfect. We have 2 more questions. Congrats, several congrats from the Diana Jerusalinsky and from Paula Lunardi. Okay, other question is from Paula Lunardi. So apparently the profile of microglia adapts better than astrocytes during the aging?
I think they both act together, if I understood well the question.
It's about the adaptation of microglia better than astrocytes. Do you think that is the situation?
Probably yes. We have to define this. There are very few studies about senescent microglia. There are much more studies on microglia in aging, this phenotype known as dystrophic microglia. But we must define what is the senescent microglia and how they interact with other cell types.
Okay, I will do the last question, and we keep a couple of questions for the last discussion, for the end of the discussion. This is from Carlos Humberto Andrade. Congrats on the lecture. I was wondering what period of time can senescent cells be considered ashash, and mean at what point can cells be considered senescence?
This is a good question. It's complicated to answer this question in a short time, but I think that the senescent cells, the senescent phenotype is very well described to know. I mean, it's not one single phenotypic feature, but several phenotypic features in addition to the mitotic arrest, right? But it is considered that senescent cells are accumulated during aging in almost every single tissue, but also during development, senescent cells play a real important role in shaping the development and the organ shapes and functions. So this could be the large answer. Thank you.
Thank you, Dr. Barbeito, for your answers, for your presentation. I am going to introduce you to Dr. Andrea Cristina Paula Lima. She is researcher at the Biomedical Neuroscience Institute of University of Chile, associate professor at the Medical School of the University of Chile, PhD in Chemical and Federal University of Rio de Janeiro, Brazil, and postdoc fellow University of Chile. The leader of her conference is R and R2 partakes in the mitochondrial dysfunction caused by amyloid beta oligomers. Welcome, Dr. Paula Lima.
Thank you very much, Patricia. Thank you all for being here. I would like to thank the organizers for the opportunity to show our work. I will present a set of results to convince you that the isoform 2 of the ryanodine receptors, or RYR2, partakes in the mitochondrial dysfunction caused by amyloid beta oligomers. I am also professor of the Faculty of Dentistry at the University of Chile, besides the Faculty of Medicine. And I would like to show you some general characteristics of Alzheimer's disease. Most of you know that it's a neurodegenerative disorder, so it means that at the later stages of the disease, we have neurodegeneration, a loss of neurons, but it's important to note that this degeneration is preeminent in the hippocampal region. And the hippocampus is the region of the brain that is responsible for memory, and Alzheimer's disease is a disease characterized by memory loss, progressive memory loss. But we have also some histopathological hallmarks, which are the neurofibrillary tangles and senile plaques. This is the triad that makes Alzheimer's, I mean degeneration, tangles and senile plaques, amyloid and tau. Senile plaques, neurofibrillary tangles are formed by hyperphosphorylated tau protein inside the neurons. They are intracellular aggregates, but senile plaques are otherwise extracellular aggregates of several components, but the main ones are the beta amyloid peptides or amyloid beta peptides aggregates. And we know that these aggregates are related to synaptic failure, the loss of communication among neurons, and to this memory loss that we observe. And these lesions are first observed in the hippocampal region, but it also spreads for the cortex with the aggravation of the disease. And the amyloid peptide that is present in these plaques also have another conformation that is known as oligomers, that are soluble conformation that can affect neurons before neurons die. How is this? If we treat amyloid hippocampal neurons, primary hippocampal neurons, with amyloid beta oligomers, and you measure calcium signals, the intracellular calcium signals using an indicator such as fluorophore, you see that there is an increase in calcium concentration inside the cells. And this increase is in a particular form. We will see in the next slide the quantification of this. You see that this increase is not a very huge increase, is about 2 times the basal line, but it stands for a long time. It's a long-lasting signal that will determine some defects in cells. And in this graph here, you see that calcium is the same second messenger that can signal in cells to make different processes. I mean, if you have high intensity and high frequency calcium signals, it's related to long-term potentiation and memory processes. But if you have low intensity and low frequency calcium signals, such as those ones that we observed after adding amyloid beta oligomers, what we observe is that this is related to long-term depression and forgetfulness, the opposite process. So the kind of signal that is observed, the calcium signal observed inside the cell, will determine what cells will do. Okay. And we have several results that show that these calcium signals that are invoked by beta oligomers are related to several other processes, such as ROS generation in the mitochondria. So I will show details of this. Mitochondrial fragmentation, I will put emphasis on this. And also astrogliosis, I will not talk about this today, I don't have time. And spine remodeling, I don't have time either, but Dr. Cecilia Hidalgo showed a lot of results on this matter yesterday. And also on the defects in memory loss that are associated with Alzheimer's disease. I will show just a few results on this. But going back to calcium signals, what are the molecular entities responsible for the generation of the signals in neurons? We know that there are several proteins involved on this. Most of them are voltage-gated calcium channels or agonist-gated calcium channels that are on the membrane. But I would like to emphasize that the receptors that are present on the endoplasmic reticulum, which is a very important reservoir of calcium inside the cells, are very important in signaling. Why? Look at these receptors, ryanodine receptors, besides being a channel that releases calcium from the reticulum to the cytoplasm, it is also activated by calcium. So the idea is that the calcium that enters via one of these receptors that could be, for example, NMDA receptors, can activate the reticulum via RYR to release more calcium to the cytoplasm and maintain or disrupt the signaling depending on the way they do this. Also, we have IP3 receptors that are linked to this mechanism, but it's activated by IP3. And we have another molecular components that are important, such as the pumps that puts calcium outside the cells or to inside the reticulum, such as SERCA. And these calcium homeostasis will regulate a lot of important processes. I will show you some regulation of the mitochondria that is very important. Because this calcium can enter the mitochondria, and also this calcium can regulate gene expression in the nucleus and regulates for sure synaptic plasticity. And I would like to show you this scheme from Dr. Michael Berridge from 1998 that is very, very interesting to show that the concept of the reticulum could be considered a neuron within the neuron. A neuron within the neuron because it's everywhere. You see the reticulum in red here in the dendritic spine, but also around the nucleus. And this presence everywhere is very important to signal. This is an immunofluorescence staining that we made in the lab of RIR, in green, and you see that this is in fact what we observe, that ryanodine receptors are everywhere from the spine to around the nucleus. But the other thing important about this receptor is that it can be regulated by the state of oxidation of the cell. Okay, and here we see that there are several proteins that can be redox modified and have their activity controlled by redox state. But if you look the fractional open probability of the ryanodine receptor, this huge protein that is in the reticulum, you see that in the normal condition, a partially oxidized one, you see a kind of distribution of the signal that's a Gaussian. You see that there's a concentration that activates, it's open, and then in a higher concentration, it closes the receptor. But what we see is that if the cells are oxidized with tonus, oxidized tonus, is that even in low concentrations of calcium, you can see the opening of the receptor, and you don't see the closing of receptor even in the higher concentrations of calcium. And on the other hand, if you have the cells reduced, the environment is reduced, even in higher concentrations of calcium, you don't see the opening of these receptors. So the ryanodine receptors could be considered redox sensitive proteins that will respond differently depending on the state of oxidation of the neurons. They have systems that I don't have time to explain, but they have systems that are regulated by redox. And going back to neuronal cells, if we use again an indicator such as fluo4 and activate directly the receptors with its pharmacological agonist, that is for CMC, you see a very huge increase in the cytoplasm of calcium mediated by the release of calcium from the reticulin by ryanodine receptors. But if we pre-treat neurons with an antioxidant such as N-acetylcysteine, which is the major precursor for glutathione in the cells, we see that this calcium increase mediated by ryanodine receptors is very, very reduced. Also, if we treat the neurons with A-beta oligomers, that we see that increase the calcium signals in the cells, the previous presence of NAC, N-acetylcysteine, in the culture also reduces the levels of increases of calcium that we see in the cytoplasm. So you can regulate the activity of the receptors by an antioxidant. The other thing I would like to show you is that there's a proximity, a local proximity between the reticulum and the mitochondria. You see, this is a immunofluorescence in skeletal muscle by other group, in which you can see clearly that they are very close together. And this proximity favors the transfer of calcium from the endoplasmic reticulum to the mitochondria. What you see here is we transfected neurons with inverted mitoperican protein, which decreases fluorescence when binds to calcium inside the mitochondria. Okay, so we are measuring calcium inside the mitochondria. And when we treat the neurons with A-beta oligomers here in red, you see an entrance of calcium inside the mitochondria, the same we see when we treat neurons with the agonist of the ryanodine receptors, showing that the ryanodine receptors activation will directly influence calcium inside the mitochondria. And on the other hand, if we block ryanodine receptors with ryanodine in inhibitory concentrations or with N-acetylcysteine, you don't see this entrance of calcium inside the mitochondria. Okay, and as a consequence of the entrance of calcium in the mitochondria, we know that there is the Ross generation. So if we measure now with mitosox dye superoxide production by the mitochondria, what we see is that if we either use the CMC agonist of RIR or A-beta oligomers, we see an increase in the generation of superoxide, which is completely blocked if we block pharmacologically the ryanodine receptors. And the consequences of this entrance of calcium in the mitochondria and this increase in ROS generation in the mitochondria is the fragmentation of the mitochondrial network that we have already published in 2011. But it's important to note that this fragmentation that we observe in about 50% of neurons when we treat them with amyloid oligomers is completely prevented if we previously treat neurons with the antioxidant N-acetylcysteine. So the fragmentation of the mitochondrial network induced by beta oligomers is prevented with the antioxidant NAC. And more directly, when we used an antisense oligonucleotide antisense against the ryanodine 2 isoform, we have 3 isoforms in the brain, but I am showing the results that show that is the 2 isoform that is very, very related to this calcium entrance in the mitochondria. What we see is that the entrance of calcium in the mitochondria that you now are seeing as an increase in fluorescence, because now we are using ROD2 indicator, is completely blocked if we use this oligonucleotide directed to the isoform 2, and the same we observed in the fragmentation, the protection of the fragmentation that amyloid oligomers cause. And finally, there's another aspect that is very important, is that besides the mitochondria, this calcium that is mediated, calcium signals that are mediated by ryanodine receptors also influence what happens in the nucleus in terms of gene expression. And it's also related to the protein synthesis that relies on spinal remodeling. When we measured nuclear calcium signals specifically to see what happens if we don't have the ryanodine 2 isoform by using now an shRNA, again, to downregulate its expression, and using a GCAM protein directly to nucleus, and we are seeing now just nuclear calcium signals, what we see is that when we use a stimulus, such a high frequency field stimulation to activate neurons, you see nuclear calcium signals. But in the presence of the shRNA to downregulate these receptors, you see that these nuclear calcium signals are very, very much reduced. And I don't have time to show you all the results, to go through the results, but we have some published papers that describes results showing that calcium release mediated by redox sensitive RYR2 channels has a central role in hippocampal structural plasticity and spatial memory, and that N-acetylcysteine prevents the spatial memory deficit in the redox-dependent RYR2 decrease displayed by a model in which we inject amyloid oligomers directly in the hippocampus, and also that RYR2-mediated calcium release elicited by neuronal activity inducing nuclear calcium signals, Krebs phosphorylation, and a PAS4 and RYR2 expression changes synaptic plasticity. And as a summary of what we think is that amyloid oligomers activate in some way the entrance of calcium via NMDA receptors, which activate RYR to release more calcium, which enters the mitochondria, which increases ROS generation, fragments its network, and also it changes the transcription of several genes that depends on nuclear calcium signals. And that all these mechanisms could be prevented if we modulate this oxidation state of the cells. So A-beta oligomers produce redox and calcium-dependent functional alteration in the red hippocampus, leading to RYR2-mediated mitochondrial dysfunction, synaptic plasticity defects, and memory loss. I would like to thank the people who did the work, mainly Pedro Lobos, Ignacio Vega, and Alex Cordova, who is already outside the lab in a postdoc, and past members and also collaborators, especially Dr. Cecilia Hidalgo, Dr. Tatiana Lasman, Dr. Jose Luis Valdez, who participated in the work. And thanks also to the funding agents. Thank you very much for your attention.
Dr. Andrea, thank you very much for your beautiful presentations. We have not yet questions on the chat, but I would like to understand, what do you think about the role of the calcium channels regulated by the MDA, for example, in the regulation of calcium in the nucleus? How is the interaction of the relationship? Yeah.
Well, no, NMDA receptors are very important. We think there's a step-by-step flux of calcium. You must have the entrance of calcium to activate the ryanodine receptors. And if we block NMDA receptors with MK801, for example, you block everything, you know? You block calcium signals inside the cell, so you don't activate ryanodine receptors at all, okay? But if you have just NMDA receptor activation and the ryanodine receptors are blocked, what we see is that you have just a very small peak of calcium, you know, that we believe that is not sufficient to signal what cells must do in that time.
Okay, thank you very much for your explanation. Okay, we have some questions on the chat. Congratulations by Maria Daniela Santi and Carlos Humberto Andrade. Okay, then Natalie Arnold, thanks for your talk. Do you know if other methods like CUPA could activate ryanodine?
Ryanodine receptors are very, very specific for calcium, but you have some other regulators of the receptor. The main one is ROS, as I told you. If you have ROS, you regulate the opening of the receptors, but there are also other proteins such as FKBP1 that regulates the ryanodine receptors and activation. But copper, I don't believe that has a very important process, very important function in regulating ryanodine receptors at all. At least as far as I know.
Okay, thank you. Okay, great talk by Diane. And say, is NAC activated in the long-term memory deficits?
What? I didn't understand. Let me see.
Say there is, I suppose that say that the activate antioxidants are, yeah, it's involved in the deficits of the LT memory.
Okay, the antioxidants. Yeah, we had this worry about what would do NAC without the oxidative stress. And we made the experiments. We have this paper from 2018 in Frontiers, in which we didn't see any effect of NAC alone in the memory, in the spatial memory of the animals. And also we made a very difficult experiment that was to take ryanodine receptors from the hippocampus of the rats and then put it in a planar bilayer to see what happens to the state of oxidation. And what we saw is that when you have an increase in oxidation, the ryanodine receptors performed by A-beta oligomers, we have the channel more active, but only with NAC, we didn't see any difference from control.
Okay, thank you. Next question is from Jose Góngora. Thanks for your talk. Do basal ranitidine oxidation increase during senescence? Do other compounds besides NAC prevent ranitidine over-oxidation?
Oh, it's a very good question. Dr. Hidalgo has a paper with Alejandra Arias showing that indeed aging changes the activity of ranitidine receptors, and we believe that the accumulation of redox stress, oxidative stress, will change the way that the receptors respond in aging. You should see this paper from Arias et al. 2017, in which they made a lot of experiments, LTP experiments, and they show how it changes with aging. And yes, there are other ways to modulate ranitidine receptors with antioxidants, for sure. We like N-acetylcysteine because it's the main precursor for glutathione, and it also passes the blood-brain barrier. And but there are other ones that make similar effects.
Okay, thank you very much. I have a question because on the chat there is no additional questions. For me, it's very interesting that in the MAMS area and the membranes from the endoplasmic reticulum associated to mitochondria is regulating the phospholipid metabolism, regulating the calcium concentration, and starting from this specific suborganular space, we can, of the cells, can do a different process, beta oxidations, opening and closing of the channels, autophagy, and in some way is depending on calcium. What do you think the ranitidine is rolling in this crucial homeostasis of calcium in this place? Do you know about that?
Well, we are starting to learn about ranitidine receptors in neuronal cells. We know a lot in muscle cells, but in neuronal cells, we are just starting to understanding. And what we are seeing that at least in all the processes that we studied, I mean, mitochondrial, synaptic plasticity, dendritic spine remodeling, memory, okay? They are involved in all of these processes. And also there are results from other groups that relate ranitidine receptors with reticulum stress. We didn't have any results on this matter, but there are another labs that show that ranitidine receptors are also involved on this. And I strongly believe that the importance of those receptors are very high, and we just have to learn more about because they have a strong participation in signaling in excitable cells.
Okay, thank you very much, Dr. Andrea de Lima. Okay, our next speaker is Dr. Lionel Mueller-Igaz. His career is a career investigator at the National Council of Scientific and Technological Research of Argentina, PhD in neuroscience from the University of Buenos Aires, postdoctoral fellow at the Center of Neurodegenerative Research at the University of Pennsylvania, USA. And his talk is named the Translational Abnormalities in Neurodegenerative Disease, focus on TDP-43. Welcome, Dr. Mueller.
Good morning. First of all, I would like to thank the organizers for inviting me to this symposium. Today, I would like to talk to you about our results regarding the role of TDP-43 in translational abnormalities in different neurodegenerative diseases. So amyotrophic lateral sclerosis and frontotemporal dementia are two progressive and incurable diseases characterized by different behavioral abnormalities, including motor, cognitive, and social deficits. And the misfolding aggregation of mislocalization of different proteins, most importantly TDP43, is associated with both sporadic and familial forms of these diseases. So the dysregulation of processes that are related to the physiological functions of this protein seems to be a common theme in the pathogenesis of several diseases of this kind. So it's been recognized in the last few years that ALS and FTD are two ends of a pathological spectrum, and the common molecular thread that is the presence of TDP43 pathology. So as you can see here, the vast majority of ALS cases have TDP43 inclusions, and almost half of the FTD cases have TDP43 proteinopathy. So you can see here in the spinal cord of a human patient of ALS, in brown, the TDP43 insoluble material in the cytoplasm with the clear nuclei, where in a normal cell, you can see the normal nuclear localization of the protein TDP43. So very early on, we recognize and describe this sequence, the nuclear localization sequence that drives the protein to the nuclei, and we mutated the NLS and show that in culture cells, this is enough to drive the localization of the protein to the cytoplasm. So it's a functional NLS. So next, we characterized and generated these types of TDP43 transgenic mice with the idea of evaluating if they can recapitulate different pathological phenotypes, observing these so-called TDP43 proteinopathies. So we did two types of lines, one expressing the human TDP43 wild-type protein, which is predominantly nuclear, as I said, and a TDP43 variant with a mutation DNLS that drives the expression to the cytoplasm. So this is a biogenic system. Just for the sake of time, I'm just going to say that it is driven by a CAM kinase 2 promoter that drives the expression to forebrain neurons, and the responder line has the gene of interest. And in the presence of doxycycline, this drug can repress this expression, so we can have a temporal and tissue specificity. So what we did is raised the animals in the presence of doxycycline until weaning at P28 to suppress the expression of the transgene, and then release the brake and have induction of the transgene and analyze at different time points after weaning. So we describe that these animals have very interesting phenotypes. For example, we can see here a progressive neuronal loss. We can see the hippocampus of the mice here. In the controls, you have normal dentate gyrus with a progressive degeneration, which is quantified here. But some other areas like CA1 or CA3 and even cortex are relatively more spared. And another very interesting feature of these animals is the corticospinal tract or CST integrity. So this is a bunch of axons that come from the upper motor neurons in the cortex and go all the way down to the spinal cord. And if we want to analyze this tract, we can see that transgenic mice have a severe CST degeneration, as shown here in this axonal marker. Another feature that we describe is that these animals have a profound dysregulation of transcription, being TDP43 an RNA binding protein. This is very interesting. And we can see here by Affymetric microarrays that TDP43 mice have a very different transcription of profiles and controls. As I said, behavior was very important for us, and we described that these animals have several features of these proteinopathies. We have different tests analyzing motor, cognitive, and social domains of behavior, and they have profound alterations early on, 1 month after induction of the transgene, recapitulating these diseases. So I hope I convinced you that we have new animal models that are interesting for analyzing these incurable degenerative diseases. So these animals, as I show you, they have gene expression changes, neurodegeneration, and behavioral deficit, but also we have described that there is signs of gliosis and neuroinflammation and sparse occurrence of protein aggregates. This was described in these different publications. So what about the subject of the talk? So TDP-43 can also be a translational regulator. So the evidence shown that, for example, in culture cells in HEC293, a proteomic study showed that there is extensive interaction of TDP-43 with proteins of the translation machinery. In vitro, it was reported that when you downregulate TDP-43, you can alter the global translation and yield through a scar-dependent mechanism, and this was is going to be important for in a couple of minutes in the talk. In another in vitro study in SHXY5Y cells, the expression of TDP-43 NLS showed that it colocalizes in the cytoplasm with specific members of the translation machinery like RAG1 and RPS6. So our goal was to determine in vivo, because most of these cells were done in vitro, what is the impact of the mislocalization of TDP-43 in the regulation of global translation. So, as I mentioned before, TDP-43 is an RNA interacting protein, and it regulates alternative splicing. And one of the most validated target genes for TDP-43 in this function is SCAR, also known as POLDIP3. So this SCAR protein is part of a signaling pathway that regulates translation efficiency, involving a very interesting cascade, which includes mTOR, S6, and EIF2 alpha. So it has been shown in cells that when you downregulate TDP-43, you have a shift in the isoform ratio of alpha beta SCAR, which is decreased, and this leads to enhanced translation. So we wanted to see what's going on in this aspect in our mice. So we can see here that immunofluorescence for the transgene, the human TDP-43 protein with a specific antibody, showed that in controls there is no expression, and you can see robust expression in the cortex and in the hippocampus in the cytoplasm of these cells, in these neurons. When we analyze the hippocampus of these mice, we studied by radioactive RT-PCR in collaboration with a group of Dr. Kornblit in Ifebini in Argentina. And we can see that there is an increase in the ratio of exon 3 included versus excluded transcripts. So this increased exon 3 inclusion in the brain of these mice leads to an increase in alpha beta SCAR isoform ratio, and this is consistent with a decrease in translation. And this is going to be important for our experiments using Poisson profiling and Sunset that I'm going to show you in the next slides. So as a proof of concept, we use culture cells, HEC293, to understand the role of TDP-43 in the cytoplasm regulating global protein synthesis. So we use this assay called Sunset, which in a nutshell, what it does is label nascent protein chains with puromycin and antibiotic. You can label the cells with puromycin, and the nascent chains will be incorporated in puromycin, so then you can detect it with a puromycin antibody, an anti-puromycin antibody. So then the protocol was transfecting these cells with either an empty vector or the TDP-43 and less mutant, wait 48 hours and incubate with either puromycin or the vehicle, and then perform immunocytochemistry for both puromycin and TDP-43. So we can see here that in cells transfected with the antivector in the absence of puromycin, you have normal nuclear staining of TDP-43, absence of puromycin labeling as expected, and in this control cells incubated with puromycin, again you have normal nuclear TDP-43 staining, but robust labeling of puromycin, demonstrating ongoing protein synthesis. Interestingly, when we transfected cells with the TDP-43 delta NLS in the presence of puromycin, we can see that clearly there are two populations of cells, the non-transfected with puromycin labeling and the transfected cells with cytoplasmic expression of TDP-43 that have much lower levels of puromycin. So then we wanted to quantify this cell by cell. So first, we are showing here the intensity of TDP-43, and of course, both those showing empty vector and the non-transfected population of the NLS condition have same levels of TDP-43, which is what we use for normalizing this. And the cells expressing TDP-43 delta NLS have much higher levels as expected from this signal. When we analyze the puromycin intensity levels of these very same cells, we can show here again that the non-transfected population of the TDP-43 and less condition have similar levels as the empty vector, but we have a significant decrease of puromycin labeling in the cells expressing TDP-43 in the cytoplasm, demonstrating there is a decrease in global translation. So, but we wanted to know what's happening in vivo in the brains of these transgenic mice. So we turned to a technique called polysome profiling. We use, of course, the same biogenic mice, the same timeline, and analyzing 1 month after induction of the transgene. We can see here again the robust cytoplasmic expression in the cortex of these animals. So this technique allows you to separate by sucrose gradient different populations of macromolecules, and you can see here that you have polysomes, which are translationally active, and then you have monosomes and ribonucleoproteins, which are lighter. So when you do this profile of how this separates, you can see in pink the controls and in blue the transgenic mice, that the transgenic mice have lower levels of polysome fractions. And here you can see the distribution of TDP-43 in the different fractions, and the fact that TDP-43 cytoplasmic with the NLS mutation migrates also more in the polysomal fraction. So when we quantify this, we can see that there is a decrease in the area of this ratio, the polysomal versus non-polysomal, meaning that the TDP-43 and NLS mice have less translationally active fractions. So this implies that TDP-43 in vivo show decreased translational capabilities when TDP-43 is driven to the cytoplasm. So lastly, we wanted to apply the sunset method in vivo in these animals and analyze cell by cell what's going on, because the previous studies were in an homogenate of cortical samples. So we use thick brain slices. We incubated them with puromycin or ACSF, labeled the nascent peptides, and then detected by immunofluorescence for TDP-43 and puromycin. So we can see here the staining in the absence of puromycin, with puromycin, and the transgenic mice. To have a closer look, we can see that the control mice show nuclear staining, good puromycin labeling, and the transgenic animals have cytoplasmic staining and lower puromycin levels. These indeed are neuronal cells, because we did double immunofluorescence with a neuronal marker NUN, and they are colocalizing with the TDP-43 cytoplasmic staining. So when we quantify cell by cell the levels of TDP-43 immunofluorescence, we can see again that the control cells with or without puromycin have same levels of TDP-43 expression, but of course the transgenic animals have higher expression of TDP-43. When we analyze the amount of pure methylation in these mice, we can see that in the presence of puromycin, you have a significant increase respect to the noise in the background with the non-labeled with puromycin, but importantly, we have a very significant decrease in puromycin incorporation in these cells. if instead of using the whole population of cells, we do an average of each animal, we can see that this is also the case. We have a higher expression of TDP-43 and then a significant decrease in pyromethylation in transgenic animals. So to conclude, we can say that these in vitro and in vivo results indicate that cytoplasmic TDP-43 actually decreases global protein synthesis. And of course, this is interesting because it can potentially lead to functional cytotoxic effects as observed in ALS and FTD. And study provide in vivo evidence by two independent and complementary methods for a role of this mislocalized protein in the regulation of global translation, and this may have implications for TDP-43 proteinopathies. And lastly, we think that the translational modulation trying to restore proteostasis in neurodegenerative disease can be a promising field with great potential. So I want to finish by thanking the current members of our lab and the alumni, including Santiago Charif and Luciana Lucelli here, which were the main actors in the translation story. So this is our institution. These are the people providing the FALAN. Thank you very much, and I'm happy to take questions.
Thank you, Dr. Muller, for your presentations, for your interest in research. Please do questions on the chat. Dr. Muller, what is the current studies that showed the role of this protein TDP-43 in the human brains with neurodegenerations? What is the relationship with human samples? I would like to know the detail about that. Thank you.
Sure, thank you, Patricia. So, of course, this protein TDP-43 has a lot of different functions. Most of them are related to RNA metabolism. The thing is, of course, as you know, you cannot study very much what does the human brain. So we use these cellular and animal models, but in the human brain, one of the most interesting features is that it has intracellular inclusions, right? So, but the thing is that there's a big debate about if these inclusions are protective or pathogenic, right? So I think based on a lot of the studies made in animal models and in cellular models, is beginning to get clearer that the inclusions are not necessarily toxic species, and that there might be some other processes related to the ones that we are studying. For example, this regulation of translation or specific transport of RNAs, of different aspects of mRNA metabolism that are, I think, more important for the pathogenic relationship with TDP-43 with neurodegenerative disease.
I have listened from Canadian and USA researchers that maybe the TDP-43 could be a very good biomarker because it's, could be shown in a very early moment the progression of the degeneration and the alteration of the proteinopathy. Do you know something about that?
Well, the thing is that TDP-43 it could be a very good biomarker, but it has some very important problems. The first thing, for example, is a nuclear protein. So for example, to detect TDP-43 in the CSF, samples from human patients, it would be probably much more difficult than with amyloid or tau, that there's much more load of these toxic proteins in the brains of Alzheimer's, for example. So the amount of pathology is generally lower than these diseases like Parkinson's or Alzheimer's. So in the sense that TDP-43 is a very common marker of disease in different types of proteinopathies like ALS and FTD, for example, genetic-based and sporadic cases. Most of these cases have TDP-43 pathology. But the thing is that at least my take is that the attempts to use TDP-43 as a biomarker in CSF or serum even has been very challenging, because I think it would be difficult to get a significant amount of protein to these compartments to detect early on.
Okay, thank you very much. There are some questions, and congratulations by Jaime Fornaguera, Carlos Humberto Andrade, and Diane said congrats and very nice talk. Okay, a question from Mauricio Galeano. Very nice talk, Lionel. One question I have: what about the protein stability of these variants in the NLS mutant more stable than normal TDP-43?
Well, that's an interesting question, but these different forms, like for example, the mutant NLS that we use is an artificial mutant, right? It's not a point mutation found in the patients. It's more of a proof of concept to shuttle the protein from the nuclear compartment to the cytoplasm. So they have some differences in half-life, also the point mutations have. But I think it's really important also that TDP-43 self-regulates its own messenger levels. So TDP-43 binds to the mRNA of itself and regulates its own stability at the transcription level, right? So it's very complicated, the loop that you have to have into account to see the steady state of this protein. And also because when the protein goes to the cytoplasm, these mechanisms of self-regulation doesn't work very properly, so you can express more of the protein. In the nuclei, it's very, very tightly regulated. So you cannot really increase it too much or lower it too much by normal stimuli, like external stimuli. So it's very, very well regulated, but when it goes to the cytoplasm, for example, you can alter this balance, but it's an important point.
Okay, thank you, Dr. Mueller. Thank you so much for your talk and time from Sulma. Okay, Dr. Lionel, thank you very much. Please, we all the conference, all the speakers to be together for some questions and discussions. I have some past questions for you. I have some questions, one question for Dr. Sandoval. Said something like that, okay, from Jose Góngora. Thank you for your excellent talk. Do the stats of all them show the benefits on cognitions?
Sorry? What?
If the four, don't worry. If the four stats show the same benefits on the cognitive functions.
No, I understand. I have a problem with the
okay.
connection.
Okay.
For the format.
Okay. I can repeat the question. How was brought on the chat? Do the stats of all of them show the beneficial cognitive effects?
Yes. All the strats have change in cognition. I can show you a little bit of that if you want to. It's very interesting. Maybe I can share my okay. I want to show you a little bit of that. Let me find that. My computer is failing. Just wait.
Ahora sí, se ve correcto.
Okay. Can you see that?
Yes.
Yes, okay. ¿Qué pasó acá? Es que mi computador está molestando un poco.
Now is okay, right now.
Sorry. Okay. These are changes that we found with the extracts in the Murray water maze paradigm. You can see in blue the effect of the extracts in the mice are very interesting because some cases are similar to the control group, and the characteristic of these extracts are that they have inhibitors of acetylcholinesterase. They have activity of as antioxidants to inhibit the oxidation of beta carotene in vitro, and in some cases we found a neuroprotection in vitro for these extracts. I think there are many different mechanisms involved in the brain of these mice with the mice were treated, and it's difficult to resume the effects in a simple words. We are doing at the time studies of the transcriptomics to understand all the changes that are involved in the mice at the time.
Thank you, Dr.
Okay.
Sandoval. Thank you for your complete answer. Okay, I have two questions for Dr. Barbeito. One is from Silvia Oliveira. What do you think is the role of the glial cells that show absence of replicated senescence in neurodegeneration? Show absence. Could you understood the sentence?
You should open your microphone, Luis. It's closed.
Okay, I'm here.
Was clear the question, or repeat that?
Yeah, please repeat the question.
Okay. What do you think is the role of glial cells that show absence of replicate senescence in neurodegeneration?
Well, I really don't know. I don't know if it's some study have look at this specific question. I mean, the replicative senescence could be an issue with microglia proliferation, and these cells could really develop some senescent phenotype derived to that. But all these studies perhaps have been studied in vitro, but in vivo there is little information. So I really don't know.
Okay, thank you. The other question is from Bellini, Argentina. Say, do you find any sex different in the senescence phenotype?
Very interesting. We can anticipate that, but we have not studied this specifically. But if you extrapolate from other tissues and organs, there could be differences in senescence in the central nervous system. Yes.
What do you think? What is the hormone that is going favor to this difference?
Well, all the neuroendocrine regulation that is altered during aging can regulate not only sexual hormones, but also stress hormones like cortisol, DHA, as androgens. So all these responses, neuroendocrine responses, are known to be different in between females and males, and then could anticipate a sexual difference between the senescence process in the central nervous system. But as far as I know, it has not been studied. Very interesting point.
Okay, thank you, Dr. Rabeito. Next question for Dr. Lionel is from Diane. Say, is there some difference in the TDP43 mislocations in cells coming from different sexes?
Well, I assume that Diana's question is regarding human pathology, right? Because we have analyzed mice from both sexes and we didn't find any difference, but of course this is an artificial model. But as far as I know, there has been no consistent and specific studies relating this question. It would be interesting to see if there is a difference in the mislocalization. If in general the pathology, TP43 pathology is not very different between sexes. This has been studied, but there's this kind of pre-inclusion phase that some cells have more cytoplasmic TP43, but it's not insoluble yet. And this might be interesting to see if there are differences in this parameter.
Okay, thank you. The next question from Natalia Contreras is, could TDP43 mislocations be a stress signal to cause translational inhibition? Are a signal pathway that switch this protein from the nucleus to the cytoplasms?
Well, this is a very important point that Natalia raises because I didn't have time to say this in the talk, but TDP43 has a steady state mostly in the nucleus. It's like 90% in the nucleus, but it's a shuttling protein. So it performs nucleocytoplasmic shuttling, like many RNA binding proteins. So within the physiological functions, this protein goes to the cytoplasm and transports the mRNA. And when there is, for example, stress, TDP43 can participate in the formation of stress granules, which are protein and RNA structures that are related to stop general translation during cellular stress. And when the stress releases, the translation can resume. If of course this stress is chronic, it may lead to more complicated phenotypes. So in this sense, there's a few papers that suggest that the lack of resolving of these stress granules during pathology could be the initiation of the intracellular inclusions in the cytoplasm of these cells. So this is a very important point, and is something that it could be key to generate new treatments. So it's more that physiologically the protein goes to the cytoplasm for these functions, but when there's stress or there's some mutation or some other pathological event, it could be that this transport can be altered, or the structures in the cytoplasm that are formed physiologically could be malformed or not resolved properly.
Thank you, Dr. Müller. Dr. Andrea, the calcium dysfunction regulation is a transversal phenomenon in neurodegeneration in different kind of diseases, because it's involved in oxidative stress, it's involved in the proteinopathy, is involved in the alteration in metabolisms, calcium and the alteration in the cytoskeleton and also the endosome system and exosome system. There are a lot of implications of calcium in the regulation of the different kind of processing at the cell. My question is, if we, specifically in the disease that you prefer, but mainly in neurodegenerative disease like Alzheimer or Parkinson, do you like to check what happened with the calcium homeostasis in the very early stage of the disease as indicator of the progression of this different alteration of cell process? What specific time or what specific processes do you think are more important as indicator of the diseases, of the beginning of the diseases, of the as prodromic event? I don't know if it's clear my question.
I don't know really if I understand the question, but I can imagine what are you talking about, and for sure, as calcium is the main second messenger, especially in excitable cells, we know that it's involved in several processes. What we believe is that the spatial distribution, right, and the frequency, where it is, if it's a microdominion or if it's a more global calcium signal, it will determine the different processes that will happen or not happen. What we believe is that we don't know yet why in Alzheimer's disease these calcium signals are altered. So we believe that either the alteration can make some processes that are deleterious, but also it helps that cells cannot do the right signals to do what they have to do. You know. But we cannot say which is the most important feature of the signaling. We know that it starts in the membrane, and it affects the mitochondria, affects the nucleus, the endoplasmic reticulum in the protein synthesis, and also related to stress of reticulum. But we don't know what is the point of the specificity, if it's your question, in Alzheimer's disease. But because you can see several defects that are similar in other diseases either. We don't know what is the start point. We don't have this answer. We don't well, people used to say that amyloid beta oligomers is the first one in the cascade in Alzheimer's disease, but even this is now being questioned because we know that some, for example, inflammation can influence the activity of secretases and then the production of amyloid. So we don't know what is the start point, and it's a very good question if you are asking something related to this. And I would like also to take this opportunity to answer a little bit more about one question that you made before to me about copper, right?
Yeah.
That I said it doesn't regulate the receptors directly. Also, iron is important, not by regulating the receptors directly, but by generating ROS in an indirect way. It also influences the activity of the ryanodine receptor. So, for example, if you have a process in which you have a lot of iron entrance in the cells, it probably leads to ROS generation that will affect ryanodine receptors and will generate this dysregulation of calcium that will have consequences to the cells.
Okay, thank you very much to all the speakers for the fantastic presentations, for your time, for your fantastic answers, to Dr. Sandoval and to Dr. Barbeito, to Dr. Andrea, Dr. Lionel. Zulma Dueñas, she couldn't to be here because she has had an accident, a small accident, but she couldn't to be here, and said thank you very much to all the speakers, and sorry for my absence. Thank you from me too Thank you for the opportunity to discuss with you, to us, to have the contact with all the people by the chat, enrichment this discussion and this learning about different topics related with neurodegeneration and aging. Okay, see you to the 2 p.m. Time of Argentina, Brazil, Chile, Uruguay. And see you soon. I hope have you enjoyed this symposium. Some people mentioned Bellini, say thank you for excellent symposium. Thank you very much. See you. Bye-bye.
Thank you. Thank you. Thank you, Zulma.